Hey guys, so today's the last lesson of Unit 3. We have talked about aerobic cellular respiration. We also briefly talked about anaerobic cellular respiration when we went over glycolysis. Remember, glycolysis is the first step of both aerobic and anaerobic cellular respiration. So today we're going to dive into the rest of anaerobic cellular respiration. So for this lesson on anaerobic cellular respiration, we're going to be going over when we need to use anaerobic cellular respiration. We're going to compare the two different types of anaerobic cellular respiration. One includes lactic acid fermentation, the other is alcohol fermentation. Then we're going to briefly compare and contrast aerobic with anaerobic respiration, and then review some of the uses of ATP in cells. So anaerobic cellular respiration happens when oxygen is not available. This type of cellular respiration, as we've said, also starts with glycolysis. So just remember, during glycolysis, we have a net production of 2 ATP. We also have 2 NAD+ that are reduced to form NADH. And in the end, we end up with 2 pyruvate molecules. In aerobic respiration, the ETC isn't running because we don't have the oxygen at the end of it to be that final electron acceptor. So therefore, we need to have a different method to convert those high energy NADH molecules into ATP. So essentially, we're going to transfer those high energy electrons to different organic molecules instead of going through the ETC. That process of the NADH giving up its high energy electrons is called fermentation. So bacteria have evolved a whole bunch of different types of fermentation, but in eukaryotes, which are organisms like ourselves that have nuclei in our cell, there are 2 different methods of fermentation. One is alcohol fermentation, the other is lactic acid fermentation. Both of these types of fermentation have 2 stages, and they both occur entirely in the cytoplasm of the cell. For both types of fermentation, stage 1 is glycolysis. It works the exact same way as it does in aerobic cellular respiration. And then stage 2 is the fermentation, which will be either the alcohol or the lactic acid. With alcohol fermentation, essentially we start by removing carbon dioxide from each of the pyruvate molecules that we made in glycolysis. This is going to produce acetaldehyde. The carbon dioxide is just then breathed out as a waste product. The NADH molecules that were produced during glycolysis will pass on their high energy electrons and hydrogen to the acetaldehyde, which converts the acetaldehyde into ethanol. The ethanol molecules are also released as a waste product. The whole point of this fermentation stage is to oxidize the NADH in order to get back the NAD+. So it's essentially just a way to convert the NADH that was made in glycolysis back into NAD+, so it can be used again in glycolysis. It's really just a way to recycle the NADH into NAD+, so that glycolysis can keep running. So there is no actual energy created in the fermentation stages, or in the second stages of anaerobic cellular respiration. All of the energy is made in the glycolysis portion. The fermentation portion's only job is to recycle the NADH, and that's true for both alcohol and lactic acid fermentation. So the reaction is we have glucose plus the ADP and phosphate is going to get converted into ethanol, carbon dioxide, and the ATP. Both the ethanol and carbon dioxide are waste products. The ATP is made during glycolysis. That's when we make the 2 ATP, or the net 2 ATP. And then stage 2, its only job is to make sure that the cell does not run out of NAD+, because the NAD+ is needed for glycolysis. The waste product, ethanol, is the type of alcohol used in alcoholic drinks. This type of fermentation is done by yeast and other microorganisms. This is the process here, I am going to draw this flow chart out at the end of this lesson, but essentially we start with the glucose, we get the net 2 ATP produced, we get the 2 NAD plus converted into the 2 NADH, and we make the 2 pyruvate. That's the exact same as the glycolysis that we've already learned about. And then the next step, instead of pyruvate oxidation, which you would do if this was aerobic, with this type of anaerobic respiration, we are going to remove our carbon dioxide and form acetaldehyde. We're then going to oxidize the NADH to turn it back into NAD+, which produces ethanol, which is the final product of this. Okay, lactic acid fermentation is the type of anaerobic respiration done by animals, including humans. Anytime cells need more energy than what aerobic respiration alone can provide. This usually happens during strenuous exercise when your muscles are working really hard, they're burning through a ton of energy, a ton of ATP, and there's just not enough oxygen in your blood to essentially run aerobic respiration fast enough to provide all of the energy those muscles need. So the muscles will switch over to lactic acid fermentation, where the NADH produced during glycolysis is going to become oxidized, essentially giving up its electrons to the pyruvate. That's going to convert the pyruvate into lactic acid. And once again, it's just a way to recycle that NADH back into NAD+. The formula for this is the glucose plus the ADP and phosphate turns into lactic acid and ATP. As the lactic acid builds up in the muscle cells, it will cause stiffness, soreness, and fatigue. If you've ever been like running or working out or anything like that and felt that burning in your muscles, that is a buildup of lactic acid. The lactic acid can then be transferred from the muscles to the liver through your bloodstream. And then once the exercise stops, the lactic acid will be converted back into pyruvate. And then that pyruvate can go through the rest of aerobic respiration. The conversion of lactic acid into pyruvate does require oxygen, which is why that can only happen after the exercise has stopped and you build up some more oxygen in your bloodstream. Okay, so this is what it would look like. So the glucose at the top is going to get turned into pyruvate. During that process, we make a net 2 ATP. We also have the 2 NAD plus reduced to form the 2 NADH. Those 2 NADH are going to go down. They are going to drop off their high energy electrons to the pyruvate. So the pyruvate becomes reduced into lactic acid. The NADH becomes oxidized to turn back into NAD+. So remember, that is an example of a redox reaction. Anytime one molecule is oxidized, there's always another being reduced. Whichever one is giving up the electrons is oxidized, whichever one is taking the electrons is reduced. So in this case, the pyruvate takes the electrons, so it is being reduced into the lactic acid. The NADH is giving up its electrons, so it is oxidized to turn into the NAD+. And then the NAD+ just goes back up to be reused in glycolysis. As we've talked about before, there's a lot of different uses for ATP in the body. Everything from active transport, so pumping molecules into and out of the cell, vesicle transport, which, I mean, one example that we've talked about earlier this semester is when the vesicles of neurotransmitters are released into the synaptic cleft. That's a type of vesicle transport. Making proteins, contracting muscles, heating up your body and maintaining that internal temperature, all of those use ATP. Okay, I am going to briefly sketch out essentially just these flow charts in a way that, I don't know, might make more sense, or maybe these ones are just better for you. Okay, so just to kind of go over these two different types of anaerobic respiration again, you have the type that involves alcohol fermentation. It starts with glycolysis, where we convert glucose into pyruvate. During that process, we get a net of 2 ATP produced. We also have 2 NAD+, which are reduced to form 2 NADH. Then in step 2, which is the actual fermentation step, that pyruvate is going to be turned into acetaldehyde by removing carbon dioxide. The acetaldehyde is then reduced into ethanol by the NADH. So the NADH comes, it's going to drop off its electrons onto the acetaldehyde. In that process, the NADH is oxidized to turn back into the NAD+, and the acetaldehyde is reduced to turn into ethanol. So that is a redox reaction that's happening right there. And the whole point of the glycolysis portion is to make the ATP. The whole point of the fermentation process is just to recycle the NADH back into the NAD+. Then we have the lactic acid fermentation, which also starts with glycolysis. Same thing, glucose turns into the 2 pyruvates. We get a net production of 2 ATP. That is the whole point of the glycolysis portion of fermentation. We also have the 2 NAD+, which are reduced to make the 2 NADH. Then in the second step, the lactic acid fermentation, these pyruvates are going to be reduced into lactic acid. That happens when the NADH comes and drops off its 2 electrons. So the NADH is oxidized to turn back into the NAD+, and at the same time that the NADH is being oxidized, the NADH is being oxidized, it is reducing the pyruvate into lactic acid. So once again, that is a redox reaction that's happening there. Okay, please let me know if you have any more questions about this.